Okay, good evening all, good evening, good evening. Okay friends, good evening all, good evening. So today we will start the, as I said, cell communication, cell signaling, cancer, immunology. Okay, it's a kind of a easy test only. Friends, there are total 18 to 20 questions are there. Okay, so make sure that out of 18 to 20 questions, you people has to answer at least 13 to 14 correctly. Suppose example, if we are answering 18 questions, sorry, if we are solving 18 questions, make sure that you have to answer at least 13 to 14 questions correctly. Okay, okay friends, let's start. Okay, just type start, we will go it. Just type start, we will start. Okay. Okay, let's go for the question number 1. Okay, 20 seconds. Please start. Question number 1. Five seconds more. Stop, 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 stop. Please type answer. Please type answer. Okay, everyone has given the answer wrong. See, it's a very simple question. Everyone given wrong. What's the question? Anyway, first the answer will be A. Friends, relax. Slowly analyze. What is the question given? The targeted cells for the ADA gene therapy are. How can you say that directly B lymphocyte? You are not getting the question. What's the question asked? The targeted cells for the ADA gene therapy are. You all commonly know that. Everyone look into the explanation please. See, you know very well B and T lymphocytes. What the B and T lymphocytes, if they want to be differentiated, they require an enzyme called as ADA. So you all know very well, because of the deficiency of ADA, it causes a particular disease which we call it, friends, SCID. You all know very well. Now in the SCID, what we are doing? Everyone answer my question. First, we will take the ADA gene. From where we will take? We will take the ADA gene from the healthy donor. After that, this gene we are going to introduce into a suitable vector. I told you in the classroom, most probably we will use the retrovirus as a vector. Now you only tell me where I am introducing the vector. Am I introducing the vector in the B lymphocyte? Where I will introduce the vector? Obviously friends, we are going to introduce the vector into the bone marrow, because B lymphocytes never produce the ADA gene. Please remember that. So you have to understand, bone marrow cells are the one which are responsible for the production of the ADA. So if a particular person who is not producing the enough ADA gene, so by using rDNA technology, we have introduced this particular ADA gene in the bone marrow cells, not in the B lymphocytes. That's completely wrong. Because bone marrow is the major producer of ADA. Hence the correct answer will be A. Slowly think and answer. Good, Aparjita, good. Okay, get ready for the question number 2. Get ready for the question number 2. See, all questions are seems to be very simple, but what you have to think and answer. Okay, slowly. Get ready for the question number 2. Here you go. 20 seconds. Viruses can switch from lysogenic cycle to lytic cycle via. 15 seconds more. 5 seconds more. At least one fellow I am expecting answer. At least one fellow. Stop, 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 stop. Okay, anyone give the answer? Oh, very, very good, Kavita. Very good. The answer is A. UV light. Okay. So all of you look into the picture. Sorry. Everyone look into this particular explanation. Friends, everyone pay attention. You know, right? Bacteriophage can show the two type of life cycles. One we can call it as lysogenic, and we can also call it as lytic phase. So friends, all of you look into this particular process. An environmental insult such as exposure to the ionizing radiation or ultraviolet rays can cause the integrated provirus to leave the host chromosome and follow the normal lytic cycle of the viral replication, because when the bacteriophage is exposed to the very high intensity of sunlight, it cannot show its effect on the host chromosome. That's why it can easily come out and it starts leading the normal life cycle. So that is what normal lytic cycle of the replication. Do not forget that. Hence, the question number 2 answer is A. If anyone did not understand the question number 2, you can go with the answer and you can ask me for the explanation if you don't understand in the telegram. Okay, get ready for the question number 3. Get ready for question number 3. Here is the question number 3. Your time starts now. 1 minute. One minute. Think and answer. Think and answer. 40 seconds more. All questions seems to be very easy, but they will be confused. 30 seconds more. 30 seconds more. C, D. Oh, good, very good, very good. Okay, others, everyone want to answer. 20 seconds more. Stop. Stop, stop, stop. Friends, the answer will be D. What is the question they have asked? Which of the following statement is true concerning about the natural killer cell? We all know very well. We use it to define that natural killer cells are the early responders. Early responders of viral infections as well as tumor infections also. Obviously, there is no doubt. They belongs to the T cell lineage, not at all. They are not belongs to B cell lineage. They kill bacterially infected cell. Here some people got confused. Natural killer cell do not have any effect on killing the bacterial infected cell. Suppose example, if they ask you a straightforward question, which of the following cells are involved in killing the bacterial infected cell? So most probably macrophages. Means at least they will do the process of phagocytosis first. Anyway, since the question is with respect to the natural killer cell, obviously the answer will be what, friends? D. That is, they display cytotoxic effect on the tumor cell with the help of antibody. That's why we use it to say natural killer cells plays a key role in the antibody dependent cell mediated cytotoxicity. Hence the correct answer will be D. Simple. Okay, to virus infected cell. Correct. Get ready for the question number 4. Question number 4. Your time starts now. 30 seconds. I have given a very simple questions, friends, because we can expect this, but which we always confuse. 30 seconds more. Ten seconds more. Stop. Friends, the question number 4, the answer will be C. Is it? So, what is actually chemotaxis movement? Chemotaxis movement is a kind of mechanism where particular organism, you can take a bacteria, you can take a somatic cell, whatever the organism it may be, they starts moving to find their food resources with the help of certain chemical signals. That is what exactly we can call it as what? Chemotaxis. It is the phenomena whereby somatic cells, even bacteria and or other single cell, even multicellular organisms direct their movement in response to certain chemicals in the environment. You know, right? An example, take a bacteria in case of a nitrogen fixation. So what the plant root will release? Plant root release the chemoattractants. Best example, you can take an example of flavonoids and you take an example of betanes, which are lipids. Now what is happening now? They will release these chemicals. These chemicals travels towards what? Bacteria. Then bacteria get attracted. Now bacteria start directing towards the root for the process of nitrogen fixation. So that is what we call chemotaxis. So answer both A is correct as well as friends B is also correct. Hence the correct option will be C, both A and B. Correct. Okay. Get ready for the question number 5. Your question goes here. 1 minute. Start. Your time starts now. 1 minute. Thirty seconds more. It's a very logical question, friends. Good evening, Deepika. Please continue. $15 more. 10 seconds. Stop. Stop. Anyway, the correct answer for the question number 5 is the C. Option C. Every one of you look into the given question. The question says that why the oral rehydration therapy, which requires a feeding dilute salts and glucose, is successful. This therapy saves the lives of millions of victims of cholera and other diarrhea producing diseases. What is the reason? Friends, you all know very well. First of all, what is happening in the cholera? Because of the continuous opening of the CFTR channel. What is happening? First chlorine goes out, sodium goes out, water goes out. Every single ion present in our body completely goes out. Now what is the treatment that we will do? We will just give the oral rehydration therapy or ORS supplements. Now look into the question. Now you can ask me a question. Sir, already CFTR channels are completely open. Once again, how we can send the electrolyte inside? Now because you will get it out. All this, if I given the electrolytes to a cholera patient, already channels are open. Whatever the electrolytes that we have supplied, even that may also go out. That is true statement. But what is happening is this particular Vibrio cholera only affecting active transport of glucose and sodium from the cell to the intestine. What is the point you have to remember? It is not at all affecting. secondary active transport. In a simple way, it majorly act on the primary active transport only. Since it is not affecting the secondary active transport, so whatever the ions that we have given in the form of a electrolyte or ORS, that can reach us to the targeted cell via secondary active transport. We can also call it as co-transport systems or the co-transport mechanism. So very simple question it is. Hence the correct answer will be C. Okay, get ready for the question number 6. Your time starts now. Question number 6. How do the viruses evade the host defenses? 50 seconds more. Slowly think and answer. 40 seconds more. Friends, think and answer, think and answer. 20 seconds more, 20 seconds more. Ten seconds more. Stop. So friends, the answer for the question number 6 will be B. The question asked, we have discussed, how do viruses evade the defenses mechanism? Here the word evade in the sense escape. How viruses are going to escape from our host defense mechanism? By producing enzymes that destroy the antibody molecules. That is true. I will agree with the option A. There is no doubt about it. You have to listen my statement. If a particular virus want to secrete certain substances which can kill the antibody or which can destroy the antibody, first of all, virus has to enter inside. Virus has to enter inside the cell. Once it enter inside the cell, then virus get replicated, then it produces certain proteins. Those only will go and destroy the antibodies. For that, virus has to be entered. If the virus wants to be entered inside, they have to get a hide from our immune defense molecule. Hence the correct answer is by hiding inside host cell where they cannot be detected by the host immune system. Once they get hidden, then they produce all the enzymes, leukocidins, then they can inhibit all the processes. The correct answer, most appropriate answer will be B. Okay. Okay friends, just give me one moment of time. Just a moment. Okay, get ready. So what is the question number that we are discussing? Question number 6 is over, and get ready for the question number 7. Okay, get ready for the question number 7. Here you go. Everyone, 30 seconds. Ten seconds more. 10 seconds more. Stop. Oh shit. I'm so sorry. Yeah. Everyone given the answer? Given the answer? C. Okay. Anyone else? Okay. The question says that, friends, which of the following act as the receptor for the Epstein-Barr virus on the B cell surface? So friends, first of all you have to remember on the B cell surface what kind of receptors are present. See, CD45 is not present on the mature B cell. Everyone remember, sorry, CD40 is not present on the mature B cell. They will be present on the pro B cell, or they are present on the pre B cell, not on the mature B cell. Whereas CD21 is the major receptor that is present on the B cell surface, which is acting as a major, major receptor for the Epstein-Barr virus, which is the option B, CD21. Anyway, CD23 do not have any action, even though CD40 is present. All of you remember, even though CD40 is present, it is responsible for what purpose? Signal transduction. It do not have any role in associating with this particular Epstein-Barr virus actually. Hence the correct answer will be B. No problem, no problem. Slowly think and answer. Get ready for the question number 9. See friends, from now onwards questions are very simple, but it seems to be very confused if you don't analyze it properly. Get ready for the question number 8. "20 seconds. Please go. Your time starts now. Ten seconds more. 10 seconds more. Stop, 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 stop. Stop. What is the answer? The answer for question number 8 is the B. The question says that which of the following molecules involved in calcium dependent cell adhesion? Every one of you quickly answer my question. Apart from the cadherin, everyone answer. Apart from the cadherin, what is the other molecule that is also dependent on the calcium? Everyone please answer my question. Apart from the cadherin, what is the other molecule also involved in association with the calcium? We read in the class. Anyone? See, here the calmodulin cannot be an answer. Why? Because calmodulin never associated with the cell adhesion process. Very good. So, along with the cadherin, friends, selectin. Why? What happened? Everyone forgot. Selectin is the another molecule which is always associated with the cell adhesion and with respect to the calcium. And you also know very well, cadherin will show what kind of interaction? We all know very well, cadherin will show the homophilic adhesion. Even selectin also associated with the calcium, but it is going to show what kind? It is going to show the heterophilic adhesion. Please don't forget this particular statement. Hence the correct answer will be a B. Friends, get ready for the question number 9. Question number 9. Here you go, question number 9. 30 seconds. No, for this question I am giving 1 minute. 1 minute, think and answer, friends. Think and answer. Think and answer, friends. "40 seconds more. 40 seconds more. Everyone, please think and answer. Think and answer, friends. As I already said, question seems to be very simple but very logical. 10 seconds more. I wish everyone will give the correct answer. Stop, 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 stop. Question number 9, the answer will be D, which is called as indirect fluorescence antibody test. What actually we are doing in this particular method? All of you look into this. In this particular method, see everyone, we are incubating a normal renal tissue. We will take a kidney tissue, and that kidney tissue we will take along with the patient serum. What the patient serum contains? Patient serum contains antibodies. So we have taken a kidney, a renal tissue or any tissue of a kidney, and we have taken antibodies which are present in the patient serum, and then treating with the fluorescent labeled antibody IgG. Then what is happening? Based on the color will give the which antibody is going to be associated, and color also will give the quantity also. So immunofluorescent indicates of immunoglobulin G is a deposition, and it is used as a important diagnostic tool, right? So please remember, the correct answer will be what? D. Simple question and direct question, the technique. Okay, next question. Get ready for the question number 10. Question number 10. Even though you made it wrong, there is no problem. Please try to understand the question. If you are not confident in the examination 100%, don't choose that question. Whenever you are 100%, then only go for the answer, because we are having plenty choice. Okay, question number 10. Your time starts now. Toxic shock is caused by. Question seems to be very simple but very logical, I am telling you. Thirty seconds more. Twenty seconds more. Question number 10. Someone already given option C, B, A. Most of the statements are appropriate, friends. You have to choose the very correct option, very close option. Okay. Question number 10, the answer will be C. Whomever given the option B, please try to understand. First of all, what do you mean by the word toxic shock? And first of all, toxic shock can be stimulated by what kind of antigen? You read in the host to pathogen interaction, there is a term called as super antigens. Friends, what are actually super antigens? Super antigens are the one which will stimulate the immune response more than the normal. So because of super antigens, what happened? First, T cells are going to get activated. There is no doubt about it, right? Not only T cell, even there is a production, there is an activation of the B cell also. But you have to remember, when compared to the T cells, B cells will be more rapidly activated. I am not using a word that the T cell is not going to get activated. T cell will get overactivated, but when compared to the T cell, B cell activation will be more. As a result, there is a abnormal production of the cytokines, especially, friends, interleukin 3 will be produced more in amount when compared to the normal. Hence, the correct statement will be C. Question number 10, option C. Okay, friends, please get ready for the question number 11. Get ready for the question number 11. Your time starts now. 30 seconds for this question. 30 seconds. 25 seconds more. 15 seconds more. Stop. Stop, 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 stop. No one has given answer. Anyone? Question number 11. Very good, very good, very good. Question number 11 is what, friends? B. You know, right? Phos and June are the two important transcription factor. I hope, friends, all of you remember. This is what kind of a pathway? MAP kinase pathway. Correct, na? Whenever a particular signal, first of all, EGF. Whenever EGF associated receptor get activated, upon activation of the receptor, autophosphorylation will occur. You all know very well. Upon autophosphorylation, RAS will get activated, then GRB2 get activated, then SOS get activated. Then immediately what is happening, friends? There is an activation of different kinds of protein. What do we can call RAS, RAF, MEK, ARC. Finally, the ARC, which is a kinase, enter into the nucleus, activate the two transcription factors, Phos and June. Now this Phos and June will come and associated with which gene? AP1 gene. We read it already, right? Hence the correct answer will be what? B. Very simple. Okay, friends, please get ready for the question number 12. Please think and answer the question. It's a really interesting question. Get ready for the question number 12. Here you go. 1 minute. One minute. Thirty seconds more. 20 seconds more. Fifteen seconds. Question number 12. Okay, stop. Question number 12, the answer will be C. What is the question asked? All of you focus on the given question. What could be the single best unique description of interaction of the alpha beta T cell receptor with the peptide MHC molecule? So you have to remember here, they are asking a question. You know, right? T cells having alpha receptors and beta receptors, correct na? So alpha and beta are major receptor. So the alpha and beta T cell receptor, they were always associated with what? They are always associated with the MHC molecule. We all know very well. Now the question they have asked, see, anyway, this is true statement, don't get confused. There is no doubt about when the T cell associated with the MHC, there is no doubt there is an activation of the T cell. But that is not the most appropriate statement. They are asking, give us the unique description. This is what? Friends, please understand the given question very carefully. They have asked a question, give the unique description. This is the general process. When the T cell associated with the MHC, obviously T cell is going to get activated and releasing the cytokine molecules. That's a generalized statement. It requires the peptide to be presented by the MHC class 1. There is no doubt about it. But friends, why this statement is wrong? Because in the given question, they did not mention whether it is a T helper cell. They did not mention whether it is a T cytotoxic cell. They did not mention. So if they have given either T helper or cytotoxic T cell, based on that we can tell whether the statement B is correct or not. That also we know it. So option B cannot. It cannot be a unique description. First of all, option only wrong. Why? Because they did not mention in the question which type of T cell. Now you look into the option C. It involves CDR binding to the both peptide as well as the MHC. What do you mean by that? Which means, see, all of you listen to me very carefully. Look into this. This is the T cell. Here, imagine alpha beta receptors are there. This is MHC molecule. Correct, na? So you can understand here antigen is present. That is taken by this particular cell. Correct, na? So you are able to see, right? There is an interaction. This interaction only, friends, we can call it as a what we can call it, CDR binding interaction. I am not saying that they are bind to the CDR. It is called as CDR binding interaction. Majorly CDRs are present or where? They are present in the antibody. In the variable light chain of the antibody, CDRs are present. This interaction called as CDR interaction. Always keep that in your mind. This is not a CDR region. The interaction is called as CDR interaction. Hence the correct statement will be what, friends? Option C. Simple. Get ready for the question number 13. Question number 13. Here you go. 1 minute. 1 minute. I know you will make this kind of questions wrong, friends. Look into the given question carefully. "40 seconds more." Super fast. Someone has given very fastly. "15 seconds more." Stop. Question number 13, the answer will be B. Question number 13, the answer will be B. The question says that a cell with a damaged DNA can lead to cancer if it is allowed to divide. If it is allowed to divide. If I don't want to allow it to divide, then what kind of things I have to block? The cell can get prevented from the division by blocking G1 to S phase transition and G2 to M phase transition. See, we cannot arrest directly at the checkpoint. First, we have to start with the G1 only. If there is a CDK mutation or the P53 get mutated, the cell undergo the excessive division, which leads to the cancer. So that cannot be an answer. So the answer will be the normal functioning of cell cycle. Such cells are prevented from the division by blocking the G1 and S2 transition checkpoints and G2 to M transition checkpoints. Very good. The question answer will be B for the question number 13. Okay, here you go. Question number 14. Your time starts now. 1 minute. Okay, friends, for this question I am giving you 1 minute 30 seconds. Think and answer. You have to choose correct statements. 1 minute more. Okay, friends, 1 second. 1 second. I have to give you the options. I forgot. Okay, Hold on a minute, hold on a minute. I will write the options also here. Just hold on a minute, hold on a minute. I will write the options neatly here. Option A, B and C. Option B, C, D. Option C, D followed by E. Friends, option D, E, F. Now your time starts. Take 1.5 minutes. I will give good time for this question. Make sure that you have to give a correct answer. Now Gautam, it's multiple statements. Look into it. One minute more. Before giving the answers, please check your answers. Please check your answers. Twenty seconds more. Ten seconds more. Stop. Stop. Okay, the correct answer for the question number 14 is the option A. Option A is the correct. It's a very simple. We read many times in the class. During okay, anyway, I will tell you directly. You all know very well E2F PRB complex. You know very well. I told you many times. What is the PRB? PRB is the first identified tumor suppressor gene. What is the ultimate function of the PRB we discussed? PRB blocking blocking the transcription factor named as E2F. Friends, we read many times what is the function of an E2F? E2F function is what? Synthesizing the proteins which are required for the G1 to the S phase transition. That's the unique function of the E2F. But if I want to stop the cell division, because the cell got damaged, I don't want to allow the cell to be divided, I should block it. So how can I block it? Of course, by using the PRB. E2F PRB complex is associated with the DNA but act as a gene repressor. Of course, true. PRB has to block the E2F, has to block the cell division. So obviously it is acting as a repressor. There is no doubt. Option A is absolutely right. Option B, what I said about the E2F? E2F is the transcription factor. What is its function? It is involved in stimulating the genes which are required. It is involved in stimulating of the genes which can synthesize the proteins that are required for the G1 phase to the S phase transition. Of course, option B is correct. No doubt. Look into the option C. They are asking a question. They are giving a statement. Cell loses PRB. Now you will try to understand. There is no PRB. Now no one can block the E2F. Then what happened? E2F stimulate all the genes which are required for G1 to S phase transition. It will allow the synthesis of protein. The cell easily enter into the S phase. That's what happens. Now a cell that loses PRB activity as the result of RB mutation would be expected to lose its ability to inactivate the E2F. So E2F is activated. Obviously true. Then E2F will stimulate all the genes. Then finally cell will enter into S phase. No doubt option C is correct. Now if you start looking into the option D, so A, B, C are correct. Option D, E2F PRB complex act as a gene activator. No, not at all. Why they will act as a gene activator? PRB block the E2F. It can act as a gene repressor. That's why we call it as a tumor repressor gene. It is not an activator. Option D is wrong. Option E, E2F act as a nuclear transporter. No, not at all. E2F never enter into the nucleus first of all. E2F can act as a transcription factor which is involved in the stimulation of the gene. Now, and they said that, okay, I agree with this, even though, an example, E2F can act as a nuclear transporter also, it is required for G1 to S phase. But they said that it is required for mitotic phase. No, option E is also wrong. Option F, a cell that loses PRB activity as the result of RB mutation expresses inactive E2F, which allows the cell to enter into the G2 phase. No, right? It will allow the cell to enter into S phase. Hence, friends, the correct statements are option A, option B, option C. Simple. Hope everyone understand without confusion. Okay, friends, get ready for the question number 15. Question number 15, your time starts now. For the question number 15, I will give 2 minutes of time. Think and answer. Here you go. Question number 15. I am giving 2 minutes of time. Take time. It's a very good question, friends. One minute more. You take the time. 1 minute more. Someone has given answer. Gautam. Okay. Followed by Nandini. Savita. Okay. Okay, fast. Okay. 30 seconds more. 20 seconds more. Stop. Stop, stop. The answer for question number 15 is the option C. Friends, you only try to understand what is P24. You know, right? If this is the structure of a HIV virus, it contains a genetic material RNA. And what is this one, friends? This one. This is only a protein coat which made up of P24. I want every one of you remember the statements right now, whatever I am going to say. Suppose whenever unprotected sex happened or injection of a drugs or syringes. So if we want to determine whether the person is HIV positive or not, originally our body takes incubation period is how much? 90 days. You all know very well. But if you want to know it very faster, very faster, within 10 days, we will go for a test called as a NAT test. What test? Nucleic acid test, which we call NAT test. Now you have to understand, in the NAT test, what we will observe. In the NAT test, we will observe whether whether our body contains this particular P24 antigen or not. How can I tell if our body contains P24 antigen? Undoubtedly, body starts producing the P24 antibodies against that particular antigen. That's what we can determine in this particular NAT test. It's a very accurate test. Within 10 to 12 days of unprotected sex, you can easily determine whether the person is HIV positive or not. So P24 is present in the serum. So what is that? All of you listen. Early stage of infection. Now, if a particular person having that particular P24, what the body starts doing? It starts producing the anti-P420 antibody. So anti-P24 antibodies are high in the serum, which indicates that the viral antigen is progressing, which we call viral latency. If All of you listen. Anti-P24 antibody begins to decline with the corresponding increase in the P24. What do you mean by that? P24 antigen is gradually increasing and production of P24 antibody is decreasing, which means the viral number is increasing more than the normal. Then it will take a greater time for the body to produce the P24 antibodies. So which means when the anti-P24 antibodies are going to be decreased, when the viral latency progressed continuously. Progression of HIV from latency of light means the number of P24 antigens are more than the normal and body is unable to produce the antibodies very fastly to affect on that particular P24 antigen. Hence the correct answer will be C. Very good, very good. The question number 15 answers. It's a very important question, friend. Okay, get ready for the question number 16. I hope everyone is enjoying the questions. Okay, next question number 16. Here you go. 30 seconds. Seconds. Fifteen seconds more. 15 seconds more. 10 seconds more. 5 seconds more. Stop. The answer for the question number 16 is the IFN alpha. So we commonly know, right? Which among the following inhibit the viral replication within the cell? If one cell get affected by the virus, this is the cell affected by the virus. Now this cell starts releasing the IFN alpha. This cell will reaches, this IFN alpha reaches all the adjacent cell. It will help all the adjacent cell to get protected from this particular virus. Anyway, what was that? IFN alpha. Perfect. Get ready for the question number 17. Your time starts now. Yes, here you go. 1 minute. Thirty seconds more. 30 seconds more. Twenty seconds more. Okay. Stop. The question number 17, the answer will be C. You know that RB tumor suppressor protein bind to the transcription factor E2F and it prevents the E2F not to enter into the S phase. Right? Hence the correct answer will be C. Okay. Get ready for the question number 18. Get ready for the question number 18. Okay, 1 minute. Question number 18. They are asking which is not they are asking, not. Friends, 15 seconds more. Ten seconds more. Stop. Question number 18, the answer will be A. Dash is not a characteristic feature, not they are asking. So you have to understand, friends, cancer, see, you have to understand, you know, right, this is a cancer cell releases certain factors. Now you all know very well, whatever the factors released by the cancer cell, where they will come and attaches. Whatever the release factors released by the cancer cell will come and attaches to the same cell. That is what we can call it as a autocrine signaling. Correct, na? Whatever the factors released by the cancer, it never allow the adjacent cell to become cancerous. No, that's not possible. Growth and division without stimulation by the growth factor. This is the characteristic feature of cancer. DNA damage does not halt the cell division or stimulate apoptosis, right? Replication that occurs unlimited number of times. Of course, that's the characteristic feature of cancer, which is not is A. Hence the question number 18, option A. Okay, so friends, that's all for today. And there are 2 more questions are left. So you can find the PDF in the Telegram. You can solve that remaining 2 questions also. Okay, so please kindly prepare for tomorrow. Developmental biology. Okay, that is also one of the most important topic which you have to read compulsory. Tomorrow also we will try to solve 16 to 18 questions. Okay, so I hope the discussion is really helpful to all of you. I'll see you in the tomorrow class. Okay, thank you all, friends. Thank you. I'll see you in the tomorrow class. Okay, thank you all. Thank you.